This is the North Atlantic Ocean. From the surface, it looks like most cold northern waters, gray, vast, and largely featureless. Fishing vessels work these waters regularly. Seals populate the ice margins. The ecosystem here is well documented, at least at the surface level. But descend below that surface, past the light, past the depth where recreational divers operate, down into the mesopelagic and bathypelagic zones where pressure increases dramatically and water temperatures drop to just above freezing. And you will find an animal that most people have never heard of, doing almost nothing, in complete darkness, and has been doing so for longer than most modern nations have existed. The Greenland shark holds the verified record for the longest lifespan of any vertebrate animal on Earth. Individuals have been estimated at over 400 years old. They move at less than 1 mile per hour. They reach sexual maturity at around 150 years of age, and the majority of them are partially blind from a parasite that attaches directly to their corneas. In this video, we will know everything about this shark, how it survives in such darkness, and exactly how they live for so many years. Welcome to Deep Ocean Unsolved. The Greenland shark, Somniosus microcephalus, belongs to the family Somniosidae, commonly called the sleeper sharks. The name is straightforward. These animals are slow, sluggish by most measurable standards, and largely sedentary compared to other large predatory sharks. In terms of size, they are substantial animals. Most adults fall between 2 and 4 meters in length, roughly 6.5 to 13 feet, and weigh up to approximately 1,000 kilograms, or around 2,200 pounds. Some individuals have been measured at close to 6.4 meters, or about 21 feet. They are one of the largest shark species in the world by maximum recorded length, though they are far less well known than species like the great white or whale shark. Their range covers the North Atlantic and Arctic Ocean. They are found in the waters around Greenland, Iceland, Norway, Canada, and the United Kingdom, and have been recorded as far south as the Gulf of Mexico at significant depth. They prefer cold water, between 1 and 12 degrees Celsius, or roughly 33 to 54 degrees Fahrenheit, and are capable of diving to at least 2,200 meters, or about 7,200 feet below the surface. In summer months, they tend to descend to deeper, colder water. In winter, some individuals move closer to the surface beneath Arctic ice. Their swimming speed averages around 0.34 meters per second, approximately 0.76 miles per hour, or 1.2 kilometers per hour. For comparison, the average adult human walks at roughly 3.1 miles per hour, or about 5 kilometers per hour. This places the Greenland shark among the slowest moving large vertebrates currently documented. The sleeper shark classification is, in practical terms, an accurate description. Despite this, stomach content analyses of Greenland sharks have consistently revealed a diet that includes fast-moving and large prey: Atlantic salmon, halibut, seals, porpoises, squid, and various fish species. Historical records and individual specimen analyses have also documented polar bear remains, reindeer, and in at least one case, the remains of a moose. How an animal moving at under 1 mile per hour regularly consumes prey, capable of moving many times faster, is a question researchers have spent considerable time on. The most supported current hypothesis involves ambush predation of sleeping prey. Marine mammals, unlike fish, require sleep, and when they sleep at depth, they become largely stationary. Researchers believe the Greenland shark uses its electroreceptive system, the ampullae of Lorenzini, which detect the electrical fields produced by living animals, along with its lateral line system, which detects pressure changes in the water, to locate and approach stationary prey slowly enough to avoid detection. This has not been directly observed in the wild due to the logistical difficulty of filming animals at these depths, but it remains the most plausible explanation for the available evidence. One of the more unusual features of the Greenland shark is the near-universal presence of a parasitic copepod, a small crustacean called Omatoikinesis kuki, which attaches itself to the cornea of the shark's eye. Estimates suggest that close to 90% of adult Greenland sharks carry this parasite. The attachment causes measurable damage to corneal tissue, and in many individuals results in significant visual impairment, partial or complete blindness in the affected eye. Given that the parasite frequently affects both eyes, many Greenland sharks are navigating the deep ocean with severely limited vision. This is not, apparently, a significant impediment. As noted, their primary sensory systems for hunting are electroreception and the lateral line, neither of which involves vision. In the near-total darkness of their deep-water habitat, vision would provide limited utility regardless. What makes the parasite additionally interesting is that it is bioluminescent. The copepod produces a faint biological light. Some researchers have proposed that in the complete darkness of the deep ocean, this bioluminescence may incidentally attract prey toward the shark's face, functioning in a manner loosely analogous to the lure of an anglerfish. This has not been confirmed, and the relationship between parasite and host remains an active area of study. Whether the arrangement constitutes a net harm, a neutral relationship, or something approaching mutualism is not yet fully established. For most of recorded history, very little was known about the Greenland shark's lifespan. Conventional methods for aging sharks, examining growth rings in vertebrae, similar to counting tree rings, do not work reliably for Greenland sharks because their cartilaginous skeletons calcify differently than those of most other shark species. The standard tools were not applicable. In 2016, a team led by Julius Nielsen at the University of Copenhagen published a paper in the journal Science that addressed this problem using a different approach entirely. The method relied on the biochemistry of the eye's lens nucleus. The lens nucleus is formed during embryonic development and, crucially, is never replaced or metabolically renewed over the course of an animal's life. The proteins laid down in the lens nucleus at birth remain there, chemically stable, for the entirety of that animal's lifespan. Because these proteins incorporate carbon from the environment at the time of their formation, they carry a radiocarbon signature, a measurable ratio of carbon-14 to carbon-12 that reflects the atmospheric conditions of the period in which they were produced. Nielsen's team used a known historical radiocarbon anomaly as a calibration point. Nuclear weapons testing conducted primarily in the 1950s and 1960s produced a significant and well-documented spike in atmospheric carbon-14, commonly referred to as the bomb pulse. Any organic tissue formed before this period carries a different radiocarbon signature than tissue formed after it. This allowed the researchers to establish, with reasonable confidence, whether a given shark was born before or after the mid-20th century, and to extrapolate approximate ages for older individuals based on baseline pre-bomb radiocarbon levels. The team examined 28 female Greenland sharks. The smaller, younger individuals in the sample showed the elevated carbon-14 signature consistent with post-1963 birth. The largest shark in the sample measured 502 centimeters, just over 16.5 feet. Her lens nucleus radiocarbon signature placed her estimated age at approximately 392 years, with an uncertainty range of plus or minus 120 years. The conservative lower bound of that range was 272 years. The upper bound extended past 500. The study's conclusion was that the Greenland shark has a minimum lifespan of approximately 272 years, with the largest individuals likely exceeding 400 years. This placed the Greenland shark above the bowhead whale, previously the longest-lived vertebrate on record at around 211 years, and established it as the longest-lived vertebrate currently known to science. A shark estimated at 400 years old today would have been born around 1620. That is the same year the Mayflower made its crossing of the Atlantic. It predates the founding of the United States by more than 150 years. Galileo Galilei was still alive. Most of the borders visible on a modern world map did not yet exist. This is not a rhetorical flourish. It is simply what 400 years means when placed against recorded human history. The longevity of the Greenland shark is not a biological accident. It follows from several interconnected physiological and environmental factors that researchers have identified with reasonable confidence, along with some that remain under active investigation. The most fundamental factor is metabolic rate. The Greenland shark is an ectotherm, its internal body temperature matches the surrounding water. In water hovering near freezing, all biochemical processes slow considerably. Cellular respiration, protein turnover, DNA replication, all of these proceed at a fraction of the rate they would in a warm-blooded animal or in an ectotherm living in warmer water. Because biological aging is, at its most basic level, the accumulation of cellular damage over time, a slower metabolic rate translates directly into a slower rate of aging. The cold water the Greenland shark inhabits is, in a very real biochemical sense, the source of its longevity. This is consistent with broader patterns observed across species. Among fish, the longest-lived species tend to be cold water dwellers. The rough-eye rockfish, for instance, can live to around 200 years and inhabits cold Pacific waters. The ocean quahog clam, Arctica islandica, an invertebrate, has been documented at over 500 years old and lives in cold North Atlantic waters. Cold temperature and long lifespan are a recurring combination in marine biology. In the Greenland shark's case, additional mechanisms appear to be involved. Preliminary genomic research has identified gene variants associated with enhanced DNA repair pathways and increased resistance to oxidative stress, two factors that contribute to cellular longevity. This research is ongoing, and firm conclusions have not yet been drawn, but the findings are consistent with what one would expect from an animal displaying what some researchers classify as negligible senescence, the absence of measurable biological decline with age. The shark's body chemistry also plays a role in its survival in cold water, though this is distinct from its longevity mechanisms. Greenland shark tissue contains high concentrations of trimethylamine oxide, TMAO, and urea, both of which function as osmolytes, stabilizing proteins against denaturation in near-freezing temperatures. These same compounds, however, make fresh Greenland shark flesh toxic to humans and most other mammals. TMAO breaks down during digestion into trimethylamine, which in significant concentrations causes neurological symptoms: disorientation, nausea, impaired coordination, that Greenlandic communities have historically compared to intoxication. The traditional Icelandic preparation of Greenland shark, known as hakarl, addresses this by fermenting the flesh over a period of 4 to 6 months, either buried in gravel or hung in a ventilated structure. The process breaks down the toxic compounds, but produces a strong ammonia odor that most people unfamiliar with the food find difficult to tolerate. It remains a traditional food in Iceland, particularly associated with the midwinter festival Thorablot. The reproductive biology of the Greenland shark compounds the significance of its longevity in ways that have direct conservation implications. Sexual maturity is estimated to occur at approximately 150 years of age. This is, to the best of current knowledge, the longest juvenile period of any vertebrate animal. An individual that is 100 years old is, biologically, still not a reproductive adult. Gestation is believed to be lengthy. Estimates range from roughly 8 to 18 years, though this has not been directly confirmed, and litter sizes are thought to average around 10 pups, each born at approximately 38 to 42 centimeters, or 15 to 16 inches in length. The sharks are ovoviviparous, meaning eggs hatch internally, and pups are born live. These reproductive parameters matter because the Greenland shark was commercially harvested at significant scale during the first half of the 20th century. The primary target was the liver, which can account for up to 25% of the animal's total body weight in large individuals. Liver oil was used as an industrial lubricant and fuel source. At peak harvest levels, estimates suggest approximately 30,000 Greenland sharks were taken per year. This occurred before the 2016 aging study, before, in fact, any reliable method existed for determining the shark's age. The assumption at the time was that these were simply large, slow-moving sharks, unremarkable from a longevity or reproductive standpoint. What was not known was that many of the animals being harvested had been alive for well over a century, and that females among them may have been carrying pups through a gestation period measured in years. The Greenland shark is currently listed on the IUCN Red List as near threatened. Commercial hunting at historical scales has largely ceased, but they remain vulnerable to bycatch. unintentional capture and fishing gear targeting other species. Climate change presents a longer-term concern. Arctic waters are warming at a rate faster than most other ocean regions, and the cold temperatures that define the Greenland shark's habitat and that are directly responsible for its extraordinary lifespan are gradually shifting. Population recovery from the levels of 20th century harvest is difficult to model with confidence. With a 150-year maturity threshold, the standard frameworks used for fisheries recovery do not apply in a straightforward way. The full demographic consequences of historical harvesting may not be accessible for generations. The Greenland shark is not a particularly dramatic animal. It moves slowly, lives quietly at depth, and attracts far less attention than larger or more visually striking species. What makes it significant is the accumulation of biological facts that, taken together, describe something genuinely outside the normal range of vertebrate life as we typically understand it. An animal that lives past 400 years, matures at 150, gestates for potentially a decade, and moves through complete darkness at under 1 mile per hour, is operating on a timescale and by a set of biological rules that have very little overlap with most of what we study in vertebrate biology. The 2016 paper did not change what the shark is. It gave us a more accurate picture of something that was always there, below the surface, doing what it has always done. There is still a considerable amount we do not know. The mechanics of their reproduction remain incompletely documented. The full extent of their range is not established. The long-term impact of historical hunting on current population structure is uncertain. The relationship between the bioluminescent parasite and the shark's feeding behavior has not been directly observed. These are questions that ongoing research is working to address in an environment that makes direct observation exceptionally difficult. What we do know places the Greenland shark in a category of its own among vertebrate animals. The longest lived, one of the deepest diving, and among the least understood of the large predatory sharks. The ocean, as is often the case, has been less forthcoming with information than we would prefer. If you made it this far, thanks so much for watching, and hopefully, I will see you in the next one.